All right, welcome to Science Daily Challenge Day 4. So this is the last topic for biology before we are going to be exploring chemistry tomorrow. So we're going to be going over genetics, right? Question 1 states, what is the name for the process used to create gametes or sex cells? So the answer to question 1 is going to be B, or meiosis, right? Remember, mitosis is the process where we're going from a diploid cell, so basically a cell that has 46 chromosomes, and then when that cell divides, each new daughter cell is going to also have 46 chromosomes. With meiosis, you start off with a diploid cell, and after 2 rounds of division, you're going to be producing 4 haploid cells that are basically all different from one another. Question 2, right? The cells at the end of meiosis are genetically identical. This is false, right? Because we know that the cells at the end of meiosis, they have to be very different, right? Because of the different processes that we're going to be going over in more details, such as, for example, crossing over of homologous chromosomes and various other things. The cells at the end of mitosis are, however, genetically identical. But for meiosis, they're going to be very different from one another. All right, questions 3 and 4 now are a bit of some probability. So you might be wondering, right, this is kind of like math, why is this here? Probability is something that's going to be seen pretty heavily in the context of genetics. And the 2 important rules are what's known as the law of addition or the law of multiplication. Lastly, question 5 states a pure breeding yellow plant is crossed with a pure breeding green plant. All of the progeny are green. The recessive phenotype is what? So pure breeding basically just means homozygous. And homozygous means we basically have 2 identical alleles. So for example, for the yellow plant, right, let's have letters like, we can use letters like capital Y, right, will represent yellow, and then let's say lowercase y represents green. So my pure breeding yellow plant, its genotype should be capital Y, capital Y. Whereas the pure breeding green plant is going to be lowercase y, lowercase y. And now if I cross these together, right, the resulting progeny is going to have the capital Y and a lowercase y. And so with that genotype, if you see that they're all green, right, this basically means that green must be the dominant phenotype, and therefore the recessive phenotype should be A, or yellow. And so genetics here, right, especially in the context of making things like Punnett squares, assigning genotypes, these are all very important topics that we will explore in a lot more detail once the lessons begin. But for now, hopefully this was a quick, informative introduction to genetics. Thank you.